Migle Gabrielaite. I'm a Marie Curie postdoctoral fellow at Vilnius University, and I'm working on Achromobacter species bacteria that are opportunistic pathogens associated with poor clinical outcomes and often highly adapted to patient environment, such as cystic fibrosis patients. Achromobacter Ruhlandii, the Danish epidemic strain, is extremely resistant to antibiotics and is highly adapted to patient environment. However, we don't really know how they adapted to what genetic determinants actually led to this adaptation to human environment. Therefore, I'm working on that and trying to understand it. I used almost 60 fully whole genome sequenced Achromobacter Ruhlandii isolates from Danish epidemic strain and a few dozens of Ruhlandii that are not Danish epidemic strains, trying to compare and find out what's the difference, what are the genetic determinants causing this epidemic phenotype. Overall, first thing I found out is that Danish epidemic strain is geographically restricted to Denmark and is not spread over the world, unlike other epidemic strains of Achromobacter Ruhlandii. And it seemed to be emerged in 1980s, which is consistent with the first known isolation, which happened in 1993. As you can see here from the dating analyses. Another very interesting thing we observed is that even though we know that Danish epidemic strain is highly antibiotic resistant and can be resistant to basically every used antibiotic in the clinic, its resistance seems to be coming down. And that might be because of the new innovative treatments for patients with cystic fibrosis, the modulator therapy introduced in 2020, 2021, where people started feeling better, started using less antibiotics, and antibiotic resistance is naturally going down, which might be pretty exciting. Next, we performed genome-wide association studies, to understand what are the main genetic differences between Danish epidemic strain and the rest of Achromobacter Ruhlandii. And what we found is that most of the genes, meaning 182 genes out of almost 260 that were significantly associated with the epidemic phenotype, were horizontally transferred. And 5% of these genes were related to iron acquisition, which is unusually high. Overall, there were two main gene clusters, one related with replication, recombination, and repair, and another related with defense mechanisms that were overwhelmingly significantly associated with epidemic phenotypes. And finally, one of the most interesting findings is that next to the Danish epidemic strain, which we see here, there are two other clusters of Ruhlandii that seem to also be epidemic and seem to exhibit similar genomic features to the Danish epidemic strains. So in the future, I'm planning to do thorough analysis of these isolates too, to actually get closer to answering the question, what makes a Ruhlandii strain epidemic in comparison to just environmental opportunistic isolates that we see in other cases. And with that, I would like to thank first and foremost my collaborators I work with, also European Union for funding my project through Marie Skłodowska-Curie program, and Vilnius University for giving me this opportunity to be a part of a scientific community there. And thanks a lot.